Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal taking you all to the next part of Biomolecules. आज हम लोग प्रोटीन का स्ट्रक्चर, उसका स्टेबिलिटी एंड व्हाट आर द डिफरेंट टाइप ऑफ प्रोटीन अलोंग विद देयर सिग्निफिकेंसेस सीखने वाले. जैसे ही आगे बढ़ेंगे तो हमारे पास फर्स्ट थिंग इज द स्ट्रक्चर ऑफ प्रोटीन. अगर मैं प्रोटीन का कोई भी स्ट्रक्चर कंसिडर करता हूं तो सबसे सिंपल से लेके कॉम्प्लेक्स स्ट्रक्चर तक हम लोगों को चीजें पढ़नी. फर्स्ट दैट इज वेरी वेरी सिंपल स्ट्रक्चर कॉल्ड एज प्राइमरी स्ट्रक्चर. अब ये प्राइमरी स्ट्रक्चर एक्चुअली है क्या? इट इज अ चेन ऑफ अमीनो एसिड. अमीनो एसिड का चेन है, बट ये चेन कैसा है? जस्ट अ स्ट्रेट चेन. So this is a straight chain. कैसा चेन है? बस स्ट्रेट चेन. अब ये स्ट्रेट चेन का स्टेबिलिटी कैसा रहेगा गाइस? जितना चेन को हम लोग सीधा रखेंगे उतने ही इंट्रामोलिकुलर इंटरेक्शंस क्या हो जाएंगे? कम हो जाएंगे. यहां पे द चेन डज नॉट have intramolecular interactions. Now, what do you mean by intramolecular interaction? Such interactions which can be taking place within the same molecule itself. All the interactions which are taking place within the same molecule itself by different groups is nothing but your intramolecular interactions. So the interactions taking place within the same structure through the set of different molecules. Through the set of different molecules. Now, if I'm considering the stability of structure due to less intramolecular interaction, the stability is very, very less. However, there is a presence of peptide bond in throughout the structure of your primary structure of protein. So there is presence of peptide bond, but this is least stable structure. Out of all the structure of protein, this one is least stable structure. So now, moving on to the next structure part of protein, that is secondary structure. If I am considering the secondary structure, there are two basic models of secondary structure. So there are two models for secondary structure. If I am considering the first model, in which a straight chain, that is the primary structure of protein, is spirally coiled in a helical fashion to form a helix-like structure, a spring-like structure. So whenever the first case is called as alpha helical structure. Now, what do you mean by alpha helical structure? This is spirally coiled primary structure of protein. Primary structure of protein. Now the protein is going to get folded or coiled within itself. As soon as the folding starts, the molecules are coming close together and they can show intramolecular interactions as well. So there is the folding of structure occurs, and it generally take place inside your rough endoplasmic reticulum. as the lumen of rough endoplasmic reticulum contains some enzymes which are needed for the folding of this particular primary structure. Now, as soon as the folding increases, the foldings are going to help in two cases. First one, it reduces surface area of the protein. Now, as we have to fix this particular protein inside the small area, then we have to reduce down the surface area as well. And the foldings are basically doing the same. So they are helpful in reducing down the surface area, while the next case is basically they allow intramolecular interaction to occur. So what is this intramolecular interaction? If I'm considering the coiling of this particular structure, in this case, some electronegative element. Now, what do you mean by electronegative element? That is, the elements which are rich in the electrons. So they are basically having a large number of electrons inside them. So whenever the electronegative elements are coming close to hydrogen, they are going to form a superficial bond called as hydrogen bond. And that is going to increase the stability. The hydrogen bond is basically represented as dotted line, and you can see this particular structure, this dotted line as the hydrogen bond. So whenever oxygen or nitrogen or sulfur or fluorine, some of these elements, whenever they are coming in close contact with hydrogen, they are forming hydrogen bond. So they are forming H bond, but the H bond formed is within the same structure itself. That's why this type of hydrogen bond is called as intramolecular hydrogen bond. So in this particular case of alpha helical structure, there is going to be the intramolecular hydrogen bonding taking place. So what is the advantage of this particular bonding is basically it is increasing the stability of alpha helical structure. So it increases the stability of structure. It increases the stability of structure. Now, if I'm considering R groups of amino acid, you can see in the particular structure that R group are always exposed outward. Can you see this? Okay. So in this case, R groups of every amino acid in secondary structures, specifically alpha helical, R groups are exposed outward. Now, what is the use of particular R group? Whenever they are exposed outward, they are reducing the steric hindrance within the molecule. Whenever the R groups are exposed outward, they are reducing down the complete steric hindrance, that is excessive stress, or the space required is basically reducing down inside a structure. Now, what does that mean? If I'm considering a structure like this of alpha helix and R group is always exposed on inner side. So if R group is bulkier, if R group is like a sumo, then this 2 sumo cannot fit inside a particular structure. So basically, whenever R group is taken to be exposed inward, then the steric hindrance will increase, and that will decrease the stability of structure. That's why R group are always exposed outward in case of alpha helical structure. So moving on to the next part, that is beta pleated sheet. So there is beta pleated sheet. So now what are this sheet? These are formed by stacking of primary structure all together on each other. So there is stacking of primary structure of proteins. So whenever we are stacking all the structure or arranging them on each other continuously, so there is like a set of papers. So whenever we are considering the beta pleated structure, it look like a set of papers, a set of papers arranged on each other, arranged on each other. So what is it going to play a role? So whenever we are stacking all the structure, they are increasing the stability by forming intermolecular hydrogen bond. If I am considering this as my primary structure and the other one is like this, so there is hydrogen bond taking place within this two particular structure. So whenever the bonding or molecular interaction taking place within two different set of structure, that is called as intermolecular interactions. So in this particular case, the stacking of molecules, the stacking of molecules is helping the set of structure, the set of structures to interact with each other through intermolecular hydrogen bonding. Like as we see in case of alpha helical structure, that was intramolecular hydrogen bonding. So in this case of beta pleated sheet, it will be intermolecular hydrogen bonding. The hydrogen bond is formed between two different primary structure of proteins. So whenever there are two or more primary structures stacked over each other with intermolecular hydrogen bonding, such structure of protein is called as beta pleated sheet. So whenever I am considering the beta pleated sheet structure, this can also be folded, but at some extent. This can also be folded, but at some extent. Why? Because the peptide bond is opposing the turning of this particular structure. Whenever I am considering alpha helical or beta pleated sheet, both structure can be turned or fold at certain extent itself. So the folding of this structure forms a hollow area, like a cylinder. And through this particular gap, the molecules can be exchanged across the membrane. So whenever the molecule either has to go inside the membrane or outside the cell, they have to cross through a cell membrane. And this crossing is possible because of this hollow gap present inside the protein structure. So the gap acts as channel for transferring the molecules, and this is how the beta pleated sheet structure are helpful in cell membrane category. So moving on to the next set of structure, that is tertiary structure. Now whenever I am considering tertiary structure, this can either be formed by multiple primary structures. Multiple primary structures are basically folded to form a woolen ball like structure. So whenever the primary structures are basically folded on each other to form a woolen ball like structure to increase the intramolecular interaction, so which is again increases intramolecular interactions, such structure is basically called as my tertiary structure. So what if we are trying to make a tertiary structure using your secondary structure? Is that possible? Yes, it is possible. So whenever I am considering two or more secondary structures, which are joined together, what are they going to do? They are joining together either by ionic bond or covalent bond. Easy. So there is bonding taking place between two or more secondary structure to form a tertiary structure either through ionic bond or covalent bond, and the structure can be like this: alpha beta alpha, alpha alpha beta, beta beta, or alpha alpha structure. So there are various possibilities to form a tertiary structure. If I'm considering alpha beta alpha, then two alpha structures are joined together by a beta structure as well. Or I'm considering alpha alpha beta, then there are two alpha structure neighboring to each other, and then the beta pleated structure is attached to it. So whenever I'm considering a tertiary structure, it's not just a structure made up of primary, but it can also be made up of secondary structures as well. So it's not that difficult, but tertiary structure, whenever I am considering, it helps to increase stability. Yes, there is hydrogen bonding, but along with that, there is disulfide bond as well. Except for primary structure, hydrogen bond and disulfide bonds are present over there in a protein structure to increase the stability. And what does this disulfide bond means? That means two sulfur groups are attached together by one bond, and this bond is known as disulfide bond. So disulfide bond is formed by an amino acid called as cysteine. So cysteine amino acid is responsible for formation of disulfide bond. Is that clear? So next one is quaternary structure. So if I am considering quaternary structure, then the structure which can be formed by more than two tertiary structures, or multiple numbers of secondary structure. Such structure will be called as quaternary structure, and this is most advanced structure of a protein. So if I am considering the quaternary structure, then for this case we can consider one example as hemoglobin. So hemoglobin, which helps in carrying oxygen within a body, which helps to carry oxygen within a body and present inside our red blood cells. Okay, so this hemoglobin is a molecule which is present inside red blood cells and helps to carry oxygen in body. So that is your quaternary structure. Now why is it quaternary? Because hemoglobin is a complete structure made up of 2 alpha chains and 2 beta chains. So there are 2 alpha and 2 beta chains are involved in a complete structure of hemoglobin, and that's why it is called as your quaternary structure. So this is a part of structure, guys, from a protein point of view. If I am talking about the types of protein, then there are basic 3 types of protein. So first one is simple proteins. So what does it mean by simple protein? So whenever I am considering simple proteins, these are made up of just, this just is really important, just amino acid. So there is no other group or any enzymatic action or any synthesized amino acid involved in the formation of simple protein. So all the amino acid are natural amino acids. Now if I am considering the conjugated protein structure, the conjugated structure is basically whenever I am adding some other molecule along with the protein. So there is a major component. A major component is always going to be a protein, but there is some minor component which is going to be added on a protein. So it can be some other molecule. We will figure it out what other molecules are going to attach on a protein to form a conjugated protein structure. So next case is derived protein. So whenever I am considering a name derived, what I will remember is that it is derived from something, right? So whenever the proteins are deriving from something using either enzymatic action, so protein which are obtained from enzymatic actions, or using synthetic amino acids. So there can be either use of synthetic amino acids or they can be derived by your action of enzymes. So moving to the simple proteins. So as the name suggests, they are actually simple in structure. So whenever I am considering a simple protein, the proteins are involved with peptide bond, and a chain of amino acid. Now this is not a primary structure of protein, guys. So the structure can be anything. It can be tertiary, it can be quaternary, it can be secondary, or it can be primary. But the chain is completely made up of amino acid. And this amino acid chain can get folded on each other. So there is, the folding is still possible. The folding of chain can take place. So whenever I am considering the folding can take place, means the stability of this particular structure can also increase. So based on the structure, based on the structure of protein, the stability of protein, is decided. Now, how does the stability is going to be decided? If the structure is quaternary, then the stability is more than that of tertiary, than that of secondary, than that of primary. So least stability is of primary and the maximum stability is of quaternary structure of protein. But now, whenever we are folding this particular structure, will it fold continuously? No, it will fold. It will fold at some extent. Now, whenever I am folding a particular protein till some extent, so what does this mean? That there is something which is opposing the folding of protein. Now, what is going to oppose? If you are twisting someone's hand, it will oppose at one point where it will start hurting. So, according to protein, whenever I am trying to fold the particular structure, this particular structure will break if more folding will take place. So during that point, your peptide bond opposes the folding of peptide bond opposes the folding of protein. Now what will something comes in your mind whenever I am talking about simple protein related to organelles? Whenever I am considering which organelles are involved in formation of simple protein, which organelles are involved in formation of simple protein. So first organelle that should come into your mind is ribosome. Yeah, without ribosome, a protein structure cannot be formed. This is the protein synthesizing unit. But the folding of protein is going to take place inside which organelle, guys? That is RER. That is rough endoplasmic reticulum. But can simple protein enter inside the Golgi apparatus? What do you think? If I am considering a simple structure of protein, then this should not, the majority of the protein should not enter inside Golgi apparatus. Why? Because Golgi apparatus was modifying a protein by conjugating them with certain groups. It can able to add carbohydrate, it can able to add lipid, it can able to add phosphate. So there are different groups which can be added on a protein structure whenever they are entering inside the Golgi apparatus. So that's why Golgi apparatus is not the case of organelle, which is majorly involved in the simple protein formation. What is the next type of protein? That is conjugated protein. So if I am considering conjugated protein, that means the proteins are attached to some other molecule. So whenever I am considering some other molecule is coming and attaching on a protein, then that can be first of all carbohydrates. If I am considering carbohydrate as my some other molecule, then they are going to form glycoproteins. And majority of the glycoproteins are involved in the case of antibodies. So they are involved in the formation of antibodies as well. In case of glycoprotein, the majority of the structure is completely made up of protein, as the suffix suggests. The suffix is made up of protein and the prefix is glyco word. So glyco is basically a minor carbohydrate group attached on a major structure of protein to form a case called as antibody. So if I am considering the next case, that is proteins attached with lipids, so they can be called as lipoproteins. Sometimes protein are attached with inorganic molecules. So now if I am considering inorganic molecules, that can be just an element. So that case will be of hemoglobin and myoglobin. So these are globin proteins. So whenever I am considering the globin, they are attached with some of the groups. If I'm considering myoglobin, the myo suggests muscles, and this particular molecule is present inside the muscles. While hemoglobin contains iron group. So this hemoglobin, heme, is representing my iron, that is Fe. And this Fe is important for attachment of oxygen. Attachment of oxygen. And this oxygen is then transported inside the complete body to every tissue in the form of just a molecular oxygen with the help of hemoglobin. So this hemoglobin is present inside your blood, while myoglobin is present inside your skeletal muscles. The muscles which are attached on skeleton or bones, those are called as my skeletal muscles. And myoglobin is one of the main components for transferring extra oxygen to the working skeletal muscles. So next case will be of some fluorescent protein. Fluorescent proteins. So whenever I am considering fluorescent protein, these are basically used in research field. Now what is the use of fluorescence protein? Whether the gene is expressing to give a protein, okay, whether the gene inside a DNA is expressing to provide a protein, such protein are useful for the visual effect. There are some researches which are taking place in which the protein formation is completely governed by a frequency of sound. So whenever I am clapping one time, the mouse color changes to red. Whenever I am tapping twice, the mouse color will changes to blue. So depending on what is the frequency of sound, that is also going to decide the expression of protein in this case. So this fluorescence protein are of different type, that is green fluorescence protein, red fluorescence protein, blue fluorescence protein, and yellow fluorescence protein. So there are four basic types of fluorescence protein which are used majorly inside the research field. So this is a part of conjugated protein, guys. Next one is derived protein. Whenever I am considering the derived protein, that means the protein has to be formed by something acting on it. So whenever I am considering a molecule of protein, whenever you are eating some proteinaceous diet, it's not like that your body is completely breaking down protein into amino acid at once. It cannot do that. It has to go in a part-wise manner. And whenever a protein is broken down using enzymes inside a stomach, it is going to form peptides in the form of peptones and proteases. So the proteases and peptones are nothing but your peptides itself. They contain a peptide bond, but they are a part of protein structure, which are not in three-dimensional structure right now. So they start to break down and form a different structure of protein in case of your derived protein. So this derived protein can be formed by enzymatic action first, or whenever a protein is formed using synthesized amino acid. Now considering an example 1 as your synthesized amino acid is selenocysteine. As the name suggests, there is some element called as selenium. What does it there? That is selenium. So if I am considering your cysteine amino acid, how does it look? That is carbon attached with carboxylic acid and amine group and hydrogen along with sulfur group, right? And this sulfur is then converted inside your selenium molecule, and this is nothing but my Selenocysteine. But this selenocysteine is not a natural amino acid, but it is synthesized amino acid. So these are the part of derived protein, which is easiest one, but tedious to form because the enzymatic action and the synthesized amino acid need to be arranged in a proper manner. So whenever I am considering any structure of protein, whether it can be primary, whether it can be secondary, whether it can be tertiary or quaternary structure, along with your two cases of secondary structure, that is alpha helical and beta pleated, we are going to use Ramachandran plot. So Ramachandran was scientist, Ramachandran plot, which is completely based on the angle between bond present in carbon and carboxylic acid and the bond present inside your carbon and NH2 and your peptide bond, that is NHC double bond. So these bond angles are basically helpful in deciding the density of protein inside a Ramachandran plot, which is helpful to decide which structure the particular protein is having, whether it is primary, whether it is secondary, tertiary or quaternary structure. So I hope you are well versed now with your structure of protein and the types of protein. Now next case is significance. The proteins are playing very significant role inside a body. First of all, whenever there is the completion of energy requirement from carbohydrate, then the use of fatty acid is also done. Then finally the proteins are going to come. So protein can provide and act as energy source. But when after depletion of carbohydrate and lipids, as soon as carbohydrate and lipids are reducing down in number and they are providing energy, then and then only the proteins are undergoing breaking down and they are going to provide energy. So what is the next case? That is, proteins are integral part of membrane. So integral part of membrane. So on membrane, they are basically embedded inside it. So whenever they are embedded or fixed inside the membrane, they can act as pump, they can act as channel, or they can act as your transporters. So there are different cases of protein whenever we are considering that in case of membrane, and this is completely helpful for transfer of molecules across cell membrane. So whenever I am transferring the molecule across cell membrane, there is a help of integral membrane protein. So if I am considering the next set of significance, the proteins are very, very important inside the muscle. So they are present in muscle building. So to strengthen the muscle, they are basically used as metabolic agent. So there is use of protein inside a metabolism for formation of new muscles. So whenever the muscle strand are broken down and they have to regain or regenerate the muscle fiber inside a body, then the protein metabolism is one of the important case which is going to be used. If I am considering protein as an hormone, is there any hormone which is made up of protein? Yes, there is. Whenever the hormones are required a receptor to work, whenever a hormone is required receptor to work, the hormone that requires receptor to transfer signal, those hormones are mostly made up of proteins. So if I'm considering example for this hormone is insulin. Now insulin is basically made up of just 51 amino acids. So it is a very small protein which is made up of just 51 amino acid. But this insulin goes and binds to one specific receptor called as GLUT4 receptors. So there are some specific receptors for transferring of glucose inside a cell. So this GLUT4 receptors are specifically working for transferring of glucose. So as soon as your insulin binds to GLUT4 receptor, this GLUT4 sends internal signal. What is internal signal? The signal which is sent within the cell after attachment of insulin on a receptor. That is called as internal signal to opens the gate of channel for movement of glucose. Channel for movement of glucose. So there will be transportation of glucose across the cell membrane is taking place whenever the insulin, which is a protein hormone, binding to a receptor called as GLUT4 receptor. This GLUT4 is very important name as per the NEET point of view is considered, and insulin is your proteinaceous hormones, and all protein hormones required a receptor for working. Is that clear, guys? So next part is there is one more significance, that is antibodies. Now as we can consider antibody as my defense mechanism. I hope everyone has taken vaccine once in a life. So whenever we are taking vaccine, we are forming antibodies inside a body of an organism or within itself or within ourselves. So whenever the antibodies are formed, these antibodies are fighting against foreign particle invading within our body. So this acts as defense mechanism by fighting against foreign particles invading in our body. So they are basically such proteins which are used for a fighting mechanism. They are defending our body from the foreign particles. We will be figuring out all the antibodies type and how they are going to function against the foreign particles in the part of immunology itself. So immunology is very interesting topic, guys, where we are going to use this particular concept of protein and their interactions for foreign particle clearance. So whenever we are considering the next type, that is known as collagen. Now this collagen protein is basically present in intracellular matrix. So it is present in intercellular matrix. So whenever I am considering intercellular matrix, means it is present between two cells and it is helping for strain to strengthen themselves. So there is, it increases the strength between two cells, or it is going to increase rigidity in that particular area. So basically, to strengthen up something, the collagen is going to be used. The collagen is very important protein for strength purpose. All right. Next case is of enzymes. Now all enzymes which are basically acting as biocatalyst. What does this mean? That is, they are going to fast up the rate of reaction taking place inside the biological system. So whichever the chemical reaction taking place, that is speed up by this particular molecule called as enzyme. And these enzymes are all proteins. So all enzymes are proteins inside the nature. So all enzymes are having the same nature as protein. So if we are considering one example of this, we can consider it as trypsin. Now this trypsin is interestingly made up of protein. So this is proteinaceous in nature. Proteinaceous in nature. However, it is going to break down protein itself. So trypsin is basically helpful for breaking down of protein itself. So whenever I am considering the enzyme that is made up of protein and that is helping to break down a protein itself, that is coming in the category of trypsin or generally your proteases. So there are various significances of protein, guys, inside the whole biology. And if we are targeting enzymes, which is very important part of this chapter, you have to be well versed with the protein and its concept, along with their monomers, along with their structure, and along with their types and how they are working. We will be figuring out inside the part called as enzymes. So for now, that's all. I hope you love this video. And guys, please do not forget to like, share and subscribe to our channel. Thank you guys. Bye bye. See you in the next video.